So, hello students and all of you, welcome back to your own channel, right? Vedantu Science Olympiad. This is Basura also, your biology master students. Now, this is the third lecture of your genetics. In the first lecture, we covered all the basics as well as your mono-hybrid cross, right? The entire mono-hybrid cross was covered in the first lecture. It was covered in the first lecture. And in the second lecture, we started off with your dihybrid cross. We also finished something called as chromosomal theory of inheritance, as well as your entire non-Mendelian genetics, such as your polygenetic inheritance, as well as pleiotropy. So pleiotropy was covered in previous class, polygenetic inheritance was covered in previous class, students. Now, students, from today onwards, right? From today onwards, we will be covering the last part of the chapter, that is called as the diseases, right? There are multiple diseases which we'll be covering in today's class. Now, disease को छोड़ के ना यहाँ पर हम start करेंगे sex determination से. Okay, students. So with that being said, students, let's start today's amazing class. That is the last class of the genetics. Okay, students. Last class of genetics. Now, students, I want all of you to understand what exactly is sex determination. Now, sex determination is a method where the one organism, right, one organism plays a major role in deciding what is going to be the sex of the offspring. Now, for example, humans only, you consider humans. In humans, it is generally male or female, students? Tell me, in the case of humans, is it generally male or female which decides the sex of offspring? All of you know, males have something called as XY chromosome. Students, yes or no? Males generally have something called as XY chromosome. And here, this X chromosome along with this X chromosome can lead to a female here, yes or no? And this X chromosome and if this Y chromosome can lead to a male. So it is generally, in the case of humans, right, it is the males, right, it is the male which decides. It is generally the male which decides the sex of the offspring in the case of humans, in the case of human, students. Now, students, when we talk about sex determination, there is one scientist which all of you need to know, called as Henking. Because of this Henking scientist, right, the entire sex determination as we know right now is possible. Now, students, in sex determination, it broadly classified into male heterogamety and female heterogamety. Remember the example I gave you about humans? The human example is actually part of your male heterogamety. Hetero means what, students? Hetero means different. Gamete means gamete. So in the case of male heterogamety, it is the males which have two type of gametes. So as you can see, students, here in the case of humans, XY in the case of male, and also in the case of grasshopper, it is the males which have X0. So two different types of chromosomes for male, and based on which is present, it can be a male or it can be a female. So here it is generally male heterogamety. Males have two different types of chromosomes and males will decide the sex of the offspring. Now, but in the case of female heterogamety, students, understand, in the case of female heterogamety, it is generally the females which decides the sex of the offspring. Here you can notice, students, it is the females which have two different types of chromosomes here. See, in the case of birds, the females have ZW. In the case of butterfly, it is again the females which have Z and Z0 here. So, female heterogamety, females have two different types of chromosomes. Male heterogamety, males have two different types of chromosomes. Now, that is called as male and female heterogamety. Now, students, apart from this, there is the most interesting type of sex determination. Now, the most interesting one is actually found with the honeybee, students, with the honeybee. Students, this is the most interesting one, so please understand this very well. Okay. Now, students, in the case of honeybee, it is called as haplodiploidy. Why? Understand here. Now, let's take a male honeybee here and a female honeybee. Okay. Students, remember, female honeybee is diploid always, while the male honeybee is always haploid. In this case, students, here 2n is equal to 32. So half the number of chromosomes are present in the case of male. Now let's say the male is producing the sperm here and the female is producing the egg here. egg and sperm done. Now, what is the process generally, students? See, here these gametes are going to be haploid again. Haploid gamete. Now, which process do you think will happen here? Anyone? Students, in this case, ask it out. Okay. In the case of here, male is both are haploid. So generally here, not generally, mitosis has to happen here. And diploid to haploid, it is always meiosis. So we have the gametes, students. We have the gametes right now, students. Okay, we have the gametes here. Now let's say this sperm and this egg will fuse right now. They'll fuse. What do we get? We will get the zygote. Yes or no? We get the zygote. And students, please remember this very, very, very well. This zygote will always and always and always be diploid. And since this zygote is diploid, this zygote will always and always develop into a female. Always develop into a female. So, as a result, students, as a result of fertilization, in the case of honeybee, it is always the female itself. You will always get the female as a result of fertilization. You will never get a male here. Never get a male as a fertilization. Now, this female can eventually become a queen depending upon the food. If she gets royal nectar, she becomes queen, or we can become a worker. Students. Both possibilities are there. So, students, this is how it's going. Okay, students, ask your doubt. No, what is your doubt? Ask your doubts. Ask your doubts. Now, male and female. Now you might be wondering. See, this is how we get female. Then how do we get male? Do we not? Then how do we get this male here? How is it possible to obtain male in the case of honeybee? Is it even possible? Yes, it is possible. So, students, in the case of honeybee, what happens to understand? In the case of honeybee, this egg is here, right? Egg. This egg will directly develop into a male drone. Male, that is your drone. Directly develop into a honeybee. And this process, right? Since honeybee, male is always haploid, so it will be haploid. Here, egg is also haploid. So, what do you call this, students? We call this process where a gamete can directly develop into a organism. Where a gamete can directly develop into organism, this process is called as what? Parthenogenesis. Parthenogenesis. So, with the help of parthenogenesis, a gamete can develop directly into a drone. So, that is how males are produced. That's how males are produced. Okay. Students, there is one thing interesting, a very interesting concept here. Okay. Now, students, tell me, all of you in the chat, this drone here, right? Okay, let me write. Okay, right here. 1 second, students. 1 second. One second, students. Okay, we'll write here. Okay. Now, students, what is interesting part here is this male right here, students. Does it have a father here? All of you in the chat, tell me. This male here, does it have a father? No. It will always have a mother here. This male will always have a mother. So, can I tell there is no father here? There is absolutely no father here. No father. Okay. Now, students, tell me, in this case, this male and this female, when they fertilize, do we get female always? We always get a female. So, can I tell this male will never have a son? Okay. There is always no son at all. So, there is no father and there is no son at all. So, in this case of sex termination, there is no father to this male and there is no son for this male here. But, students, tell me, can this female eventually will produce an egg this egg will undergo parthenogenesis and develop a male? Yes or no? This female will give an egg. This egg will develop into a male. So students, can I tell right now, this male does not have a father, but this male right here, does it have a grandfather here? Grandfather is here because there's no father here, but this female came from this male also. So there is a grandfather here. So there is a grandfather is here. Okay. Now students, this male here does not have a son whatsoever here. Does not have a son. But does not have a son here. But does it have a grandson here? Grandson? Yes. So this particular male has a grandson. So this is very peculiar right now. Here we do not have a son, we do not have a father, but we have a grandson and we have a grandfather here. So that is the sex determination in the case of honeybee, also called as haplodiploid. Haplodiploid types of your sex determination in the case of honeybee. Very interesting it is. Let's look at it. It's very, very interesting. Sex determination in the case of honeybee. Okay. Now students, 1 second. Now, with that is over, next topic is called as mutation, students. Does anyone know what is the definition of mutation, students? Does anyone know what is the definition of mutation? Anyone? What is mutation, students? Anyone can tell me? Can anyone tell me definition of mutation? Students, mutation is nothing but sudden inheritable change. There is a sudden change which can be inherited. That is called as mutation. That is called as mutation, students. Now, mutation can be mainly because of your mutation because of your genetic changes. If there is a change in DNA or change in your nucleotide, change in your nitrogenous basis, that can lead to the variations or mutation, students. Here you can see, students. So there is something called as your genomatic change, that is change in the chromosome number. Okay, this can also lead to mutation. There is a change in chromosome number can lead to mutation. For example, there is something called as aneuploidy and euploidy. Difference you understand. In the case of euploidy, the entire set of chromosomes are changed. Let's say we have 2n. 2n will become 3n. 3n can become 4n, 5n, 6n, 7n. The entire set is changing. Let's say one organism has 46 chromosomes. That is diploid condition. It can become 69 directly. Okay, triple n. Or 4n it can become 92. Okay, so that is called change in set. Change in set. Now, what is aneuploidy, students? Aneuploidy is a concept where a single chromosome is increasing or a single chromosome is decreasing here. Increase in or decrease in single chromosome is called as aneuploidy. Okay, see students, here we have hypoploidy and hyperploidy. Hypoploidy is basically decrease in chromosome. Hyperploidy is increase in chromosome. So increase or decrease in single chromosome is called as aneuploidy. A change in entire set is called as your euploidy. I hope that simple difference is clear to all of you. Simple difference is clear to all students. Now, under mutation, there are many two types. One is called as point mutation. One is called as entire frame shift mutation. Now, what is the difference, students? As the name suggests, in point mutation, there is a change in a single base pair there. See the students. Now, students, here we have TAACTGCAGGT. This is the different types of nitrogenous bases which we have studied. Okay. Now, students, imagine if this thymine is replaced by a cytosine here. A single nitrogenous base is changed. Then the entire reading will be changed. Yes or no? The entire reading can be changed that time. Okay. Now, that is called as point mutation. In the case of point mutation, a single base pair can be changed. Single nitrogenous base can be changed. That is called as point mutation. Point mutation, students. Mutation is also arise due to change in the single base pair of DNA. Okay, students. Now, what is frame shift mutation? Understand, students. What do I mean by frame shift mutation? Deletion or insertion of base pair of DNA causes the frame shift mutation. So, if I remove this entire part here, this entire frame will be changed. Yes or no? Entire frame will be changed. This, if one is removed out, and if you remove one thing, remove, we are not replacing anything. We are removing something. So if there is a deletion here, this entire frame is going to move front. Then the reading will change. Here we had TAACTG. We had CTG. Now, if this T is removed, entire frame is moved front. So in that case, we have TAA, it is same. But here right now we have CGT instead of CTG. So CTG becomes CGC. So this entire frame is only different now. So entire frame becomes different here. Can you see? Different frame. Now that is because of deletion or addition can also say. See, if you add something also, the entire frame can change. It will move back. Here the entire frame will go front. That is called as frame shift mutation. Students. And you might ask me, sir, ये सारे mutations है ना, what is the main reason for this mutation, students? There are multiple reasons. It could be diet, it could be radiation. There are multiple reasons for mutation. Multiple reasons. Okay. See, many chemicals and physical factors that include mutation, such as your UV radiation, and all of these UV radiation, all of these are called as mutagens. They're called as what? Mutagens. Mutagens are your basically chemicals of properties which can cause mutation. Mutagens. Students. Now, students, now we come to something called as pedigree. Okay, pedigree is a very important concept. Multiple times questions have come from in your examination. Okay. Now, what is pedigree? Understand, students? Not dog biscuit. Okay, not dog biscuit. In pedigree, what happens is that, right? In pedigree, it is a process by which we try to identify if there is a disease in the entire family tree. Okay, see, analysis of traits in several generations. So we identify traits, we look at entire family tree, and we try to identify certain traits which are very common in all the different generations. Okay, so analysis of traits in several generations of the family is called as pedigree analysis. Inheritance of a particular trait is represented in the family tree over generations. Let's say blue eyes, great grandfather had blue eyes, son has blue eyes, the son has blue eyes, their son has blue eyes. That blue eyes has been carried forward in every generation. Okay, in human genetics, it provides a strong tool to help you tracing out the inheritance of specific trait, abnormalities or diseases. Now, students, let's take some disease is present in great grandfather, and you know the same disease is present in your father also because of tracing. There is a very high chance that you also might get the same disease. Yes. So before you get the disease, you do something called as your pedigree analysis, and we identify the disease and we take a remedy for it. We basically cure the disease or we treat the disease before it becomes something bigger. Okay, students, in pedigree analysis we use different types of symbols. But in examination, they can ask you the symbol also directly. They can ask you, what does this symbol means? What does this symbol means? What does this symbol means? So you have to be very clear. And especially this symbol is very famous. I don't know why. This symbol is very famously asked, even in NEET examination also. That is consanguineous mating symbol here. This one, consanguineous mating symbol right here. Okay, students, in pedigree, they'll ask you to identify which type of pedigree is it. We have something called as autosomal dominant, autosomal recessive. We have sex-linked dominant, sex-linked recessive, and Y chromosome. Right? So how do you identify a particular type of pedigree? Now for that, students, I have given you a small hack, or what do I say, small tricks to identify. Here are the students, here are they. So how do you identify autosomal dominant? We can see it does not skip generation. Affected parents can have unaffected children here. And recessive, students, how do you identify autosomal recessive? It will skip generation. Here unaffected parents can have affected children. So if you look at these common features, you can identify which of the following is autosomal, which of the following is sex-linked. Similarly, if you look at the different tricks I've given here for autosomal also, for sex-linked also. Sex-linked dominant, you can see, if it is X-linked dominant, disease can never be transferred from father to son. Never will go from father to son. All daughters of an affected father will have affected daughter also. So these are different tricks I've given you. Go through this once. Try using these tricks in exam, in your when you're solving questions. Then see how helpful they are, students. Very helpful they are. Very helpful. These tricks. You can see here X-linked recessive also is a small trick here. That is, more males are affected here. In X-linked recessive, disease will always go from mother to son, father to daughter. Not other way around. Father cannot give the disease to son here. Not possible. So this is how the flow goes here. In the case of Y-linked, only males, obviously. In the case of Y-linked, only males are generally affected here. Only males are affected here. In the case of Y-linked. So students, use this following tricks to solve your pedigree questions. In NCERT, they have given you two pedigrees. Just look at that. These are two pedigree given here. This one is your autosomal dominant. See, does not skip generation. This one is autosomal recessive. It is skipping generation here also. Skipping generation completely here. That is autosomal recessive. Okay. This one is an example of myotonic dystrophy. Example. This one is a sickle cell anemia. See, sickle cell anemia example here. Sickle cell anemia example. Okay. Now, students, now we come to last topic called as genetic disorders. There are multiple disorders which can be asked to you randomly also. Okay. Now all these genetic disorders can be broadly classified in two categories. One is called as your Mendelian, one is called as non-Mendelian or chromosomal disorders. Now under Mendelian we can divide, students. Yes, these are the students. These are sufficient. These are sufficient notes, right? Now under Mendelian we can see autosomal and sex-linked. Autosomal dominant recessive. Autosomal disease, there is only one disease for your syllabus generally. That is myotonic dystrophy is an example of autosomal dominant disease. Autosomal recessive disease is PKU, that is phenylketonuria disease, SCA, sickle cell anemia disease, and thalassemia, students. Don't worry, I will teach all the diseases one by one to you. Okay. In sex-linked we have color blindness and hemophilia for sex-linked. Two are there. Okay. Now in chromosomal we have chromosomal disorder, autosomal we have Down syndrome, sex-linked we have Klinefelter syndrome and Turner syndrome. Klinefelter and Turner syndrome, students. Okay. Students, I will give the notes. Students, I will text the person and I will upload all the notes at once. All the notes I'll give you one by one now. Okay. So this coming week you can see all the notes in the description. Okay. Now, students, this is very important. Sometimes they can ask you, tell me which type of disease is PKU. You can tell me, sir, PKU disease, there's phenylketonuria disease, is a autosomal Mendelian recessive disorder. You can tell like that. Tell me what is a color blindness disease. Color blindness is a, students, color blindness is a X-linked Mendelian disorder. X-linked Mendelian disorder. So you can tell like that. Color blindness, students, color blindness. Now let's understand what is color blindness, students. Let's understand what is color blindness. In color blindness, what happens is that, students, in the case of color blindness, we can see things, right? We can see things because of the cones and rods in our eyes. Now these cones and rods get affected and an individual, a human, cannot tell what is red, what is green. They cannot tell only. They can only see gray color. They cannot differentiate colors now. Color blindness. Okay. It is a sex-linked recessive disorder. Obviously, you can see here also. Color blindness is sex-linked recessive disorder. Okay. Defect in either red or green cone of the eye. Unable to discriminate between red and green. So they cannot tell what is red and what is green, students. Okay. Okay, third lecture of biology. Okay, don't worry. It will take a lot of time. Biology is very vast. Slowly you can go ahead. Okay. Students, before you watch these lectures, please try, students, please try to watch cell cycle first. Cell cycle, cell the unit of life. Because if you do not understand those chapters, this chapter is very difficult to understand them. If you do not understand what is biomolecules, this becomes very difficult. So please start those chapters first, basic chapters. Like if you want to understand what is mitosis, meiosis, watch cell cycle first. Okay, then good. Okay, students. Now this is color blindness, unable to discriminate between red and green color here. Now why is it happening? This entire thing is happening because defect due to mutation gene present in the X chromosome. In the X chromosome, what happened? There's a defect here, right? Because of this small mutation in the X chromosome, the person is not able to see properly. Color blindness is there. And here, students, always males are more affected compared to females because females have two X chromosomes. Females have two X chromosomes. We only have one X chromosome. If there's a defect in that, gone case. Okay, males suffer more than compared to females here. Always males suffering here, students. Okay, now students, next is called as hemophilia, students. Hemophilia is very amazing. In hemophilia, what happens is that, students, in hemophilia disease, even if there is a small cut, that person will keep on bleeding. Okay, single gene, yes, they control a single gene. Okay. So photosynthesis is independent. Photosynthesis you can do on your own. Photosynthesis does not require genetics. There is no connection to genetics. Okay. Each trait can be controlled by multiple genes also. One trait can be controlled by many genes also. That is called as polygenetic inheritance. That is called as polygenetic inheritance. In polygenetic inheritance, we have many genes, one trait. Skin color, our skin color is controlled by many genes actually. There are many genes to control our skin color. Human height also. Human height is controlled by many genes. Okay. That's what I'm saying. Hemophilia is called as a bleeding disease. One small cut will keep on bleeding because the clotting protein is now produced here. Okay. Again, it's a sex-linked recessive disorder. A single protein that is part of a cascade of proteins involved in clotting blood is affected. So the whatever is involved in the clotting blood, not there. So blood is not clotted here. There is no platelet formation. Proper clotting is not done. The person will keep on bleeding. Okay. Heterozygous female carrier of hemophilia may transmit the disease to son. Heterozygous female means what? HPA, HPS. Heterozygous female. Okay. Now students, hemophilic. Hemophilic carrier. She is a carrier. This is the heterozygous female. Heterozygous female, she is a carrier right now. She can transport this X chromosome to the son. To the son. Son becomes colorblind. Son becomes hemophilic in that case. Okay. The family of pedigree of Queen Victoria shows this number of hemophilic descendants. Okay. Students, you have to remember. Unfortunately, fungus and protists, you have to remember. You have to remember slime mold. You have to remember the entire fungi, all different types of your classes of fungi, basidiomycetes, ascomycetes, phycomycetes. You have to remember. Unfortunately, students, it's biology is one subject where you have to have good memory. Concept clearing is there, but apart from concept clearing, you need to have good memory a little bit in biology, good memory, or you have to keep on revising them. Okay, students, you have to check my notes. Check my notes for fungi. In fungus, I have given one table, one large table. That table is more than enough for fungi. Just learn that one table is more than enough for fungi. Okay, students, here in the case of hemophilia, example is your Queen Victoria family hemophilia. Okay, now students. Now, what is sickle cell anemia, students? Sickle cell anemia is very, again, it is because of point mutation. Sickle cell anemia is because of a point mutation. Because of point mutation, students. Now, there is a single point mutation. Because of that point mutation, right, entire thing changes. Let's see this. I'll show you. Here, this is your normal gene. GAG is present here. GAG is present here, students. We have GAG. This is the DNA. Okay. Now, students, GAG becomes what? It becomes GTG. There is a change in a single nitrogenous base. A single nitrogenous base has been replaced now. Adenine is replaced by thymine. Students, I've told all this. I've explained what is nitrogenous base in biomolecules part. In biomoles, I've explained there. Okay. A single adenine is replaced by thymine right now. So because of that single base pair change, right, single nitrogenous base, here we get CTC, here we get CAC. CAC in the RNA becomes GAG, here becomes GUG. Because of this, again, small change here in the beta globin chain, in the peptide, in the protein, change in protein also happens. In this protein change here, in the 6th position, in normal condition, we have glutamic acid. This GLU, glutamic acid, is now replaced by valine here. Valine becomes a. Now, one small change, the entire shape of your red blood cell becomes sickle shaped. Because this one small change, RBC becomes sickle shaped. And because of this sickle shape, the RBC cannot carry enough of oxygen and the person dies. Okay. A change in the amino acid sequence caused due to the point mutation. Glutamic acid in the 6th position is replaced by the valine right now, valine, because it becomes sickle shaped. Complete sickle shaped. This is how. See, normal, sickle shaped. Everything becomes sickle shaped. And this sickle shaped cannot carry oxygen. Sickle shape cannot carry oxygen. Cannot carry oxygen, students. Okay. I should remember, usually normal is HbA, HbA is normal, AA is normal. Whenever there is a S, the person becomes carrier. A person to have this point mutation, they should have both HbS, HbS gene. They should have. Okay. Now, students. Now understand what is phenylketonuria, students. Understand what is phenylketonuria. Phenylketonuria is a very simple disease. Okay, in our body generally, phenylalanine is converted to tyrosine generally. In presence of an enzyme, phenylalanine hydrolase. Phenylalanine hydrolase, this phenylalanine is transferred to tyrosine, normal condition. But in disease condition, this enzyme is missing completely. If this enzyme is missing completely, phenylalanine is not converted to tyrosine. So in disease condition, there is no tyrosine. Because there is no tyrosine, what happens? Phenylalanine concentration increases nicely. Phenylalanine is accumulated and converted to phenylpyruvic acid. Again, very bad. So phenylpyruvic acid is more now, and this phenylpyruvic acid starts to accumulate in the joints, in the brain, or released as urine. That is called as phenylketonuria. Okay, accumulation of these in the brain results in the mental retardedness, accumulation through urine, and because of poor absorption by kidney. So that is what happens in the case of your phenylketonuria, students. Now, students, there is one more disease called as thalassemia. In thalassemia, what happens is that, students, all of you should know your red blood cells have something called as hemoglobin. Hemoglobin has 4 chains, 2 alpha chains and 2 beta chains. I have told you this in biomolecules also. There are 2 alpha chains and 2 beta chains in hemoglobin, right? Hemoglobin has, it is a quaternary structure, 2 alpha chains and 2 beta chains. In thalassemia, those chains are getting affected. Now there is again mutation here, students. See this. It is an autosomal linked disease, blood disorder resulting in the inadequate HB synthesis. There is no hemoglobin synthesis. Okay. Now, students, what exactly happens? There is something called alpha thalassemia and beta thalassemia. In alpha thalassemia, in chromosome 16, HB2A gene and HbA1 gene, both the genes get affected here. And because of this affecting these 2 genes, there is a decrease in alpha chain. Whenever there is a decrease in alpha chain, we get alpha thalassemia. Similarly, there is something called beta thalassemia. In beta thalassemia, in chromosome number 11 of the human, there is a gene called HBB gene. This HBB gene gets completely affected here, and because of that effect here, there will be abnormal beta chain and there will be decrease in beta chain here. And decrease in beta chain can cause beta thalassemia. So alpha thalassemia because of decrease in alpha chain, beta thalassemia because decrease in beta chain. That is called as alpha and beta thalassemia. Okay, decrease in chain, students. I hope all of you remember structure of hemoglobin. It has 4 chains, 2 alpha chains and 2 beta chains generally. Okay, now students, now we come to chromosomal disorders. There are 3. First one is called as Down syndrome, students. Down syndrome happens because of a extra chromosome. So students, if one chromosome is missing, very bad. One chromosome is extra in our body, then again it is very bad for our body. Okay, see, this is the karyotype here, right? If you can see on the 21st position, there's one extra chromosome here. This one extra chromosome can result in Down syndrome. Students, it's called as trisomy of 21 because 3 are there in the 21st chromosome. It occurs in both in female and male, first described by Langdon Don. These are the scientists here. This scientist told us that because of one extra chromosome in 21st position, that can lead to the Down syndrome. Now, what are the symptoms, students? What are symptoms here? Symptoms basically, students, all the symptoms are given here. Okay, first one, the affected individual is short statured, very small. They do not grow more than 3 feet or 4 feet. They do not grow taller. Okay, they have round head, furrowed tongue. Furrowed tongue means tongue is almost outside, tongue is hanging out. Okay, partially open mouth here. Palm is, this is second point here, third point, fourth point. Palm is broad with characteristic palm creases. On the hand right now, we can see a lot of lines here. Lot of lines can be seen with this kid. Okay, a lot of lines. Physical, psychomotor and mental retardedness, that is, their brain development does not happen. All of this is happening because of one extra chromosome. Imagine, just one extra chromosome, all this happens. Okay. Now, what is Klinefelter syndrome, students? Klinefelter syndrome. Klinefelter syndrome happens only in the case of males. Now, generally males have XY chromosome. In the case of Klinefelter syndrome, a male will have one extra chromosome, and because of this one extra chromosome, X chromosome extra, the male becomes completely sterile, and there are female characteristics in male. The male will start developing breast. The male will start developing breast also, and that condition is called as gynecomastia. See, students, such individual has over and masculine development. However, feminine development, that is development of breast, that is gynecomastia, also happens here. Gynecomastia, development of breast happens here. Okay, and such individuals are sterile individuals. Now, what is Turner syndrome, students? What is Turner syndrome? Turner syndrome is very simple. It happens in the case of female, and generally a female is XX, right? Yes or no? But in the case of Turner syndrome, a female is X not. That is, one X chromosome is completely missing here. One X chromosome is completely missing in this female. One X chromosome is completely gone. Completely gone X chromosome. Okay. So that is because of Turner syndrome. Symptoms is such females are sterile and ovaries are rudimentary, means does not develop, ovaries do not develop at all. Okay. And other features include lack of other secondary sexual characteristics, and the female is completely sterile right now. Sterile. Female is completely sterile. So in examination, you can get direct questions from me, direct questions like this. See here. They can ask, manifestation of phenylketonuria is given below. Choose the incorrect answer. See, mental retardedness is seen, skin rashes is seen, accumulation, blood is seen, but furrowed tongue is not seen. Furrowed tongue is seen where? Down syndrome. Down syndrome. So this question, students. Broad palm creases, visible person from palm creases happens in Down syndrome again. Down syndrome. Okay. So they can ask you such easy questions from genetics, or they can ask you pedigree questions for difficult questions, or they can ask you what is polygenetic inheritance, what is pleiotropy, or they can ask you from incomplete dominance, codominance, or they can ask you from chromosomal theory of inheritance, or they can ask you from linkage and recombination, or monohybrid and dihybrid cross. This is a very large unit. I have tried to cover the entire unit as in the shortest time possible. Right. Students, with that, I'll wrap up right now. Thank you so much, students. And next class, I will start something new chapter. I am thinking I will start with human reproduction. I will start with human reproduction in the next class. Until then, students, take care all of you. Have a great day ahead, and bye bye, students. Bye bye. Bye bye.